And this is Alex Hoischen once again. So I introduced you already yesterday. You are a colleague from Christian Gilson at the Radboud University Medical Center at the Department of Human Genetics. But you are running a different group, and this group is for genomic technologies and immunogenomics. So, and today you are talking on behalf of Bionano. At least they are sponsoring this talk because you have made a lot of experience with their technology and have generated some interesting data with it. So the title of your talk is Next Generation Cytogenetics, Optical Mapping for Comprehensive Structural Variant Detection. The stage is yours.
All right, thank you very much, Peter. I hope you can hear me and you can see my slide now.
Let me check. Yes, we do.
Wonderful. So good morning, everyone. And I have to admit, this is a really tough challenge to speak after the brilliant Hans Clevers and also Malte and Christian, who presented such nice data, but I'll do my best. My talk today will be completely different from yesterday's talk, and also really different from the talks that you've heard from the other speakers in this session. So this is about what I call next generation cytogenetics. So I guess this is in the new disease mechanism session, but it's rather new technology for rather old disease mechanisms, namely cytogenetic aberrations. But I'll dive into that in a second. Just for full disclosure, I should mention that Bionano sponsored some of our reagents. What the essence of my talk, or our own interest in optical mapping and Bionano is a very simple question. Are we ready to replace classical cytogenetics? Well, being aware of the challenge that even NGS, which I truly embrace, and a lot of us do, obviously, even NGS has not managed that yet, simply because there's some particular aberrations in cytogenetics that are really tough to detect by sequencing means. So the next challenge is up now to see whether other techniques could potentially do that. And in my very own opinion, I think optical mapping at least gets us a long way already. So if I need to explain what optical mapping can do, I try to give this illustration. So imagine you're working in a cytogenetics lab and you really master the art of karyotyping. Then you not only inspect the 46 chromosomes, chromosome by chromosome, but you really also understand the order of chromosomal bands. In essence, optical mapping is not so different from that. But now just imagine that you could zoom into each and every chromosomal bands with a much, much higher resolution. That, in essence, is what optical mapping is. So basically, each blue line you see here is a single large fragment of a chromosome, individual chromosome, with little green labels on them. So you could still consider them the cytogenetic bands, but with much, much higher resolution. And in fact, if you wish to translate, optical mapping represents cytogenetics with 500,000 bands genome-wide. And we can also measure the distance between the bands much more accurately, so that in overall we come to an up to 10,000-fold improved sensitivity. But a lot of the principles are still similar to classical cytogenetics and karyotyping. That is, that we have a genome-wide analysis, positional information, and in principle we still look at single molecules. Here's the more detailed workflow. So what we do to get our analysis going is we need a rather specific way of DNA isolation, so extremely careful way to get ultra high molecular weight DNA. So we carefully break open cells and take very long DNA molecules out. The longest molecules we can currently image are up to 2 megabases in size. Then we take that DNA and label it. Simply by an enzymatic reaction, we add a fluorescent label on a known 6-mer DNA motif. That labeled, sorry for that auto animation, that labeled DNA is then put on the Bionano chips, and it's actually linearized in the nanochannels that are on that chip. So they are small channels that only each harbor exactly one DNA labeled molecule at a time. Those molecules, after being linearized, are imaged, and then we can translate these true images into digital images that then allow to perform a de novo assembly of any given genome. And in particular here, we do that for human genomes, obviously. What we can then do based on the de novo assembly, we can just look at the expected pattern of labels in the reference. So it's the ideogram style GTG, if you wish, with ultra high resolution, and compare our de novo assembly of a given sample and see whether there's any deviation in the labeling pattern, but also in the labeling distances. And what that enables is, it enables us to call simple gains and losses, so deletions and insertions. And there the resolution goes down to 500 base pairs in size. But what is really nice, that it also enables us to find copy number changes, even repeat expansions. And importantly, and I think a lot of previous technologies like high resolution microarrays were unable to do that, this also enables us to find balanced events like inversions or translocations. So far, we in Nijmegen have roughly 20 months of experience using Bionano's optical mapping, and we have processed so far 250 samples in total. But unfortunately, we were slowed down a little bit, obviously, by the corona crisis. What we have in terms of studies, I'll present on this slide and then give you highlights later throughout the talk. So our first question is, can we use this to find known constitutional cytogenetic aberrations? Another study is not looking at constitutional changes, but somatic or acquired changes in a cohort of leukemia samples. And for both of those studies, the key question is, can we find back all the aberrations found by the standard of care, which usually is a combination of FISH, karyotyping, and CNV microarrays? Furthermore, we just started another utility test to more systematically look into the capability of detecting repeat expansions. I don't have time to show you all the data today, but that looks extremely promising as well. And finally, by having such a new technology in-house, we couldn't resist to also use it for some unsolved rare disease cases, obviously, and I'll finish with that. So first, diving into the concordance for classical cytogenetic constitutional aberrations. This is a consortium effort between us and 3 labs in France, and it's co-led by the brilliant Leila El-Khattabi from Paris. The study was set up by just including cases with known aberrations in the broader sense with developmental disease and infertility disorders. In total, we have performed this analysis for 85 samples that altogether comprise 100 cytogenetic aberrations. And you see them listed in the table below. So I would consider that more or less the daily basis or the yearly basis, or if you wish, for a cytogenetics laboratory. So all the cytogenetic aberrations that you frequently see were represented in this study. So if we perform an analysis with Bionano and do the de novo assembly and then compare to a reference genome, we usually can start the analysis by just looking at a so-called circus plot, so a circular representation of the data. So what you will see here in the outer track is just the ideogram. Then in the middle part here, you will have a CNV an SV track. All the dots represent the identified deletions, insertions, inversions, and duplications. This is followed by a CNV track. So in essence, Bionano has 2 capabilities. The core of the technology is the SV calling, as explained earlier. But on top, more or less for free, you get a CNV track that is similar to NGS, just a coverage-based method to identify mainly the large CNVs, gains and losses in the genome. And finally, in the middle here, we have a track that could display translocations. So this is a man with azoospermia. And if you look carefully, just for the CNV track, we can directly spot that this is an individual with XXY gonosomes. So a Klinefelter diagnosis was previously made and correctly identified by Bionano. Here, I just wanted to illustrate several other aberration types and just give you a first glimpse how that looks like in real-life samples. So this is a small microdeletion identified initially by chromosomal microarrays, as depicted here on the left. And in Bionano data, it looks like that. We also get a CNV call for that aberration, but also a deletion call from the SV pipeline included. We can also identify translocations, and I'll show you many more in the later talk. But what you then can see, you see a de novo assembled molecule for this patient. And this de novo assembly maps partially to one chromosome and partially to the other. So really unequivocally calling this chromosome 5 to 8 translocation in this case. And this is how that's then represented in the circus plot. We also can find balance events like inversions, and this is how they then look like. And again, we can also detect them in the circus plot. We get a lot of questions whether we also validate the entire set. I should make clear that that was not the primary purpose of this study of 85 samples. Here the question was really, can we find back the known aberrations? I think full orthogonal validation of all the calls with Bionano should be subject of a separate study that also compares it to all the sequencing methods out there to really get a glimpse. And the first effort for that is already published by the 1000 Genomes SV consortium led by Mark Chasen. But I dare to already speculate today, or base it on our data, that I think the false positive rate of Bionano seems to be extremely low. I should also add that in this particular study, half of the samples, all those that came from the France collaborators, were actually analyzed in a blinded fashion. And just to give you a glimpse of how many variants we have to interpret per each individual, by median, we find 8 aberrations, SVs that are larger than 20 kb per sample. And for the CNV calling that comes on top, mainly for the large CNVs, we usually find on average 3 to 4 per sample, of which less than 1 is larger than 1 megabase, just to give you a first flavor. So just because I only give you highlights of each of these studies now, I just wanted to mention that so far we have processed several sample types. All of them worked out fine. The entire process was relatively straightforward. But importantly, I should mention that currently it's impossible to detect Robertsonian translocations or other translocations that are balanced and break in the centromere. So there, the length of the molecules is simply not sufficient to yet identify those unequivocally, so they were not subject of this current study. However, for all other aberrations, we find 100% concordance for the known aberrations that have been identified previously by karyotyping, CNV microarrays, and/or FISH. If you're interested in more of the raw data and the entire study, I could refer you to this bioRxiv paper that we already released. Now, a similar question for leukemia. There, I think the challenge is an additional challenge that all the aberrations are acquired, so somatic of nature. But just to give you the highlights of that study, there we included 48 samples, all of which had what we think driver mutations that were acquired, and all have been analyzed previously by karyotyping FISH and/or CNV microarrays. And again, we identify 100% concordance with one little glitch. There was one sample that had 3 aberrations that were around 10% or lower variant allele fraction. Those were missed, but that was a very complex sample anyhow, of not so amazing quality. But I'll give you the details or some highlights of that study again. So here, for example, just to show you some different aberration types, you have a small deletion of the important TET2 gene identified by CNV microarrays. If you then look at just this chromosome in the circus plot, we also see that this aberration is called by the CNV profile, but also the SV algorithm. And if you then click on these, we can directly see, again, the CNV call in Bionano and the assembly-based calling of TET2, which is even more accurate, obviously, for the breakpoints. So that nicely validates even such a small single gene deletion. And it's pretty straightforward. But I should also say this is the most ideal scenario where even both algorithms basically validate each other. So that's really nice. Obviously, for leukemia, especially balanced translocations are of extreme importance. So here we have one case where we observed such a translocation. That was a known event, obviously, previously, as the study was designed such way. And this is a translocation between chromosomes 11 and 14, as already depicted in the genome-wide circus plot here. If you make a circus plot just for the 2 affected chromosomes, it looks like that. And then you just have a simple click on this translocation line, and you're guided to this plot, this is directly the zoomed in detail of the molecule of the translocation. Again, these are then the assembled molecules of the individual sample, guiding you or showing you that part of this molecule maps to chromosome 11, the other part to chromosome 14, and you can directly appreciate that this is a balanced translocation between the IGH and CCND1 locus, very well-known translocation for these type of samples. If we zoom into the breakpoint further, we can even click on this molecular map that is assembled and see individual molecules, and from that even understand how many molecules support this event and also understand the mosaic fraction for this particular case. Speaking of translocations, obviously we had several CML cases in our series, 3 exactly, and all of which presented with a Philadelphia chromosome, so the famous 922 translocation. And all of those were depicted easily, the breakpoints in ABL1 and the breakpoints in BCR. And I dare to say that the breakpoint resolution is already better than the traditional FISH methods used. Finally, in some cases of leukemia, and obviously there is very complex rearrangements that may likely be some forms of chromoanagenesis or chromotripsis and more specific. So this is one case that had a deletion of chromosome 7 and full shattering of chromosome 8. Here is zoom in just for chromosome 8. Again, if you click on that, here we can see the CNV profile and all the fusion maps detected along the chromosome. So in essence, we could now walk along this shattered chromosome and start to rebuild it to understand the exact architecture of that. I think it's a study on its own right. I think, to be precise, you may need to add some sequencing on top, but I think we get a fairly good picture of the complexity of this aberration already from the optical mapping data itself. Lastly, a new feature in the Bionano software is an area-wide CNV profile, so you don't have to look at it only in the circus plot or chromosome by chromosome, but this really resembles what a lot of diagnostic labs are using, CNV microarrays. So for the same sample, you see an AFI array on top and the Bionano genome-wide CNV profile. And here the zoom in for the 2 aberrant chromosomes, it's a gain of chromosome 12, so somatic trisomy of chromosome 12, and a partial loss of chromosome 13. And I think they're almost indistinguishable on this level. Finally, my colleagues at diagnostics said, but ideally would also identify large stretches of homozygosity in leukemia because it's an important feature for some of the cases. That is not standard yet in the Bionano software, but we just extracted all the SV calls ourselves and just looked at their genotypes. So that is irrespective of the clinical relevance of the respective SVs. And then we can just distinguish a chromosome that doesn't show any LOH and chromosome 7q that shows a large stretch of homozygosity. So we lose all our heterozygous calls and only have homozygous calls along this stretch. So I'm carefully optimistic that Bionano's technology is able to identify homozygosity. I'm not sure yet about the resolution, but I think it should be included even as a full feature in one of the next versions of the software. Just to give you some novelty in terms of research findings, what we obviously couldn't resist to do is in some of the cases we identified novel translocations, and in particular novel balanced translocations. And those could potentially lead to gene disruptions or potentially even novel fusion genes. And here are just 2 examples. So in the upper case, this translocation between chromosome 14 and 19, likely between the IGH and BCL3 genes, was missed simply because the standard of care for this CLL sample only contained microarray data and not karyotyping. So now we know that this translocation is in that sample, and maybe that has some impact for the diagnosis of this case. In the second case, the translocation itself was found between chromosome 7 and 17 by classical karyotyping. And that was the only aberration present in this individual as a somatic change. But the resolution, obviously, of karyotyping is never sufficient to dive into the details. And now we know that there's a translocation between the 2 gene loci, UBE3C and MSI2. As such, it's now interesting to follow up, ideally on RNA of this individual sample, to understand whether that is a gene disruption for one of the 2 genes or both, or maybe even a gene fusion. So just to sum up the leukemia part, we have processed 48 samples, of which 37 are simple cases with 5 or less aberrations and 11 complex cases. In summary, we identify 100% concordance for all simple cases. And again, I dare to say that there's likely a low false positive rate of overall aberrations to be interpreted. For the complex case, I already said there was one complex case in which we missed 3 of many aberrations that were just present in a low allele fraction. And there's a lot of improvements coming in terms of higher coverage. So I think in a reanalysis would even find those. Overall, for the complex cases, I should also add that a lot of complex cases are even more complex than we ever thought. And genome imaging is bringing us one step closer to the true underlying architecture. So that is fascinating as well. The next steps, at least for us in-house for leukemia, our management team of our department was enthusiastic about the first data that we presented them. So we now decided to run the full prospective study for leukemia, and we aim to start with AML, and that should start early next year. And that basically runs Bionano optical mapping in parallel to standard of care, including blinded reporting of both technologies, but also monitoring turnaround times, ease of use, and a few other parameters. And if that would be successful, we would dare to go live, especially for AML in first instance, but then it would be relatively easy to follow by other leukemia diagnoses. And I'm very impressed by the lab in Leuven, that is from Joris Vermeer's department, and Barbara de Walle is there in charge for the leukemia lab. And they are going ahead with full speed as well. And my understanding is that they will use optical mapping already in a clinical setting from next year onwards. Again, if you want to see more data of this study, I would refer you to this bioarchive paper. Lastly, a very short example, couldn't resist also for research findings. What can optical mapping do for hidden mutations in research cases? Here we collaborated with my old colleague, Roland Kuiper, who is now also at the Prinses Maxima in Utrecht. One of his interests is in so-called atypical teratoid rhabdoid tumors. So those are very rare, luckily, but very aggressive childhood brain tumors. Some of them occur purely somatic by mutations on both alleles, but in roughly 1/3 of cases, you already identify a severely predisposing germline mutation in an important gene that is called SMARCB1. However, Roland and his clinical colleagues were studying a specific family, had a strong suspicion for a germline mutation, so therefore did an entire diagnostic and research workup. So Sanger sequencing, MLPA, and diagnostic exome sequencing failed to identify a germline mutation in this gene. And in a research basis, they performed Illumina whole genome sequencing and again, couldn't find an aberration in this gene. So they contacted us, gave us cells from an EBV cell line of one of the affected individuals. We performed 300x genome coverage experiment and then just did our standard simple filtering, just looking at all the rare structure variants in this case. Here in particular, you'll see we find 12 insertions and 20 deletions. Obviously, we already had a very good candidate locus. So one of the 35 actually hit the SMARCB1 gene. So what Bionano optical mapping detected here is a little insertion of roughly 2.8 kb that created an additional label. But obviously, that is insufficient to tell exactly what material is inserted. But we were pretty sure that this is the disease-causing germline mutation in this family. So 2.8 kb insertion inserted between introns 1 and 3, that's the resolution level we get from optical mapping. So highly likely the disease caused in this family that was missed so far. And we, just to understand fully what is inserted, we tried, or Roland's lab tried several attempts to perform a PCR over the breakpoints of this event, which all remained unsuccessful. And now we understand why that is. We did whole genome hi-fi sequencing with PacBio, just with medium coverage of 8-fold, but we have 3 reads supporting this exact same event. And now we know this is actually a retrotransposon insertion in this locus, and a rather specific one, and the exact size is 2.763 base pairs. So Bionano's size estimate was actually fairly accurate. So by now we understand this element. How it really disrupts the gene is still studied on RNA level and other means by Roland's lab. But I dare to say now that we found the hidden germline variant that strongly predisposes to ATRT in this family. So just my final slide, the outlook that I see for optical mapping and the summary of our study. We so far see full concordance for all aberrations that have no balanced translocations with breakpoints in the centromeres. That remains a challenge, but honestly, my very pragmatic colleagues in diagnostics would say that for very specific diagnosis, you may continue to just look at 2 metaphases simply to count the chromosomes. For leukemia, we also receive full concordance for all clinically relevant somatic structural variants. And there I see the strongest potential to immediately replace FISH in particular, but I guess even the combination of karyotyping arrays and FISH. In research, we have the first promising approaches to find hidden structural variants. Out of 20 studies so far, 20 projects, 5 were already successful by identifying a very promising candidates. And I think, honestly, that this is a major step forward to allow what I would call a cytogenetics revolution. So it may really change the way we do cytogenetics now and actually for decades. There's new innovations coming, higher throughput is coming, as such also hopefully much lower prices per sample and higher coverage that even should enable us to identify somatic mutations below 1%. With that, I'd like to thank all my colleagues involved in this work. Main drivers of these projects are Tuomo and Cornelia from our lab. Tuomo is now back in Finland, a great postdoc, and he's setting up Bionano there. And the cytogenetic study for constitutional aberrations was a collaboration with Leila and many other colleagues in France. And with that, I'd like to thank you for your attention and happy to take some questions.
Thank you, Alex. I think you did a good job after Jansen's talk. So you kept us interested. So first question, how much conventional karyotyping are you still doing at Radboud CMU?
I don't know the exact numbers, but I think for leukemia alone, it's more than 2,000 karyotypes still. And there's still several done for infertility, recurrent abortions, some occasionally prenatally. So I think altogether it's more than 3,000 karyotypes a year.
Okay. And you foresee a longer period of transition. It will not be stopped very soon.
I mean, not immediately. I think for now, but that's very personal to our laboratory. I think the most laborious effort so far is AML diagnosis, because we always have to do the combination of karyotyping, FISH, and microarrays. So if there is a window of opportunity to improve our workflow dramatically, and as such also offer better diagnosis and even save costs, then I think AML is an excellent example. And so far, the concordance is just fantastic. So I think that will be the fastest one to integrate. And I think within half a year, we should be live with this, is my own opinion. And I hope we'll manage.
So now I look here at the questions from the audience. The first one is from Uwe Kornak. What is the detection level for somatic changes? You mentioned that a case with 10% was missed.
Yeah, so I should add that the detection level depends also on aberration type. So wherever Bionano or wherever the biology creates a unique new molecule that is not present in the reference, like in a translocation or in an interstitial deletion, there Bionano is extremely sensitive. So there I've seen newer data with up to 1,000-fold coverage or 1,500-fold coverage. So there we can go below 1% for sure. The ones that we missed are, I think, terminal deletions that are relatively small. So there we only rely on the CNV tool, and that probably is not as sensitive that it can go easily to 1%. But I think we now are much more stable at 5 to 10% already with the improvements that we saw in the software.
Okay, there is another question from Hartmut Engels. He asked, have you also analyzed translocations with breakpoints in larger sec dubs, such as the common T1122 translocation?
Yeah, I think, hey, Hartmut, great to see you, or at least hear your question. That particular one, I don't think we've done. But yes, we've looked into that both in cancer, but also in constitutional aberrations. I think in the constitutional study, there is at least 7 classical microdeletion duplication syndromes in there. All of them are detected, but for full honesty, not all of them are detected by both tools. So it's sometimes that the CNV tool is more sensitive because in the super long sec tubes, the assembly may be disrupted. But I've seen new data that even has improved. So you can subassemble only the very long molecules, and you can really bridge regions like that. And I think Joris Vermees has shown that for the very complex DGeorge region.
So the last question comes from Mark Rauschendorf. Do you think you could get to the same resolution and quality with PacBio long read sec combined with EG analysis of TADs by a high C in order to analyze structural variants and disrupted chromosomal domains? Seems this combination is already used at least for de novo genome assemblies.
Yeah, so I mean, I love sequencing, and I'm still convinced that one day there will be the one and only sequencing technology that will do everything. But I don't see that we are there yet. As he clearly mentions, you still need a combination. And while that is beautiful for research so far and for novel genomes, I don't see that that happens immediately in a clinical setting where you want to process hundreds of samples. So there, something much easier needs to happen, a much easier analysis and also easier lab flow instead of combining 3 technologies, because that's what cytogenetics is doing now. So there I clearly see an advantage so far for optical mapping. It's relatively easy. We don't need massive bioinformatics, or no, actually bioinformatician is heavily involved at the moment in-house because we rely on the software of Bionano, which has improved quite a bit. So the ease of use is probably the big argument here. And the bioinformatics complexity of all the new methods is still quite relevant. So I think we use all of those a lot, but more for research and not so much in a clinical setting yet.
Okay, thank you, Alex, for your presentation and answers to the questions.